Great. Okay, thank you to the organizers for inviting me. Over the next 11 minutes, I'll take you through some of the science behind genome editing and CRISPR-Cas9. So what I'm going to first talk about is what is genome editing, why is there so much excitement about this tool called CRISPR-Cas9, give you a brief example of how in my own research program, but others as well, are applying this to a serious genetic disease that affects millions of people worldwide, namely sickle cell disease. And then focusing on communicating clearly about genome editing versus CRISPR-Cas9, the distinction between disease versus enhancement, the distinction between somatic cell editing and germline and heritable editing, and then I'll conclude with one slide about some perverse incentives that I see that permeate how we communicate science. So first of all, what genome editing is the ability to change the sequence of the DNA in our cell with precision. And the most efficient way of doing that right now is to design a protein, what we call a nuclease, that is designed to bind to a single sequence in our entire 3 billion base pair genome, where it will sit down and cut that DNA in two. After it cuts the DNA in two, the cell recognizes that as damage to its DNA and will go about trying to repair it one of two ways. One way of repairing that DNA is to simply stitch or glue the ends back together. And in the process of stitching or gluing the ends back together, the cell can make mistakes and either remove letters of the DNA or add letters to the DNA. And this is a way of inactivating or breaking genes. The other way the cell can repair that break is if you provide another piece of DNA, it will use that other piece of DNA through essentially a copy and paste mechanism and paste the copy over the break, and thereby you can introduce small sequence changes exactly what you want. And I'm going to show you, sort of try to demonstrate that in the next two slides. So supposing you have a gene that gets decoded as the cell as a sentence that says this is a harmful genetic sequence. We can design a nuclease to break that sentence, so it says this is a harm, and on the other side, full genetic sequence. And if we allow the cell to repair that broken sequence or broken sentence, it can put the words back together precisely, and you'll end up with no change. You'll end up with this is a harmful genetic sequence. But occasionally what will happen is the cell will actually remove the full harmful. And create now a sentence that says this is a genetic sequence. Or if you look down a little bit, or it can add an extra letter. And so now you have this is a harmful genetic sequence. And so these are ways now of disrupting this harmful genetic sequence and sort of removing that from the genome. But most genetic diseases are not caused by a harmful genetic sequence. Instead, they're caused by something where you're missing it inappropriately. So here we have a gene which might be decoded as this is a sentence without a typographical error. Well, of course, that sentence has a typographical error, and that's what's causing the disease. So what we can do is make a break in that gene and then provide a template, which now in red shows it's not a full sentence itself, it's with a typographic, and the cell will then paste that sequence into the gene. Now, it doesn't do this every time. And so sometimes you get a sentence that remains unchanged. So this is a sentence without an typographical error. Sometimes you'll get that insertion and deletion. And so now you get this as a sentence with an typographical error. But sometimes you get a completion of the sentence. So it now actually says this is a sentence without a typographical error. And that's what we want to do when we're trying to repair genes. So that's the general concept of genome editing. Why the excitement about the CRISPR-Cas9 nuclease? Because that break can be made through using several different tools. But what has really democratized and revolutionized the field of genome editing is this new tool called CRISPR-Cas9, which actually is probably more appropriately called Cas9 guide RNA. Because it's a two-component system, you have one component, which is the Cas9 protein, which is the machinery that's going to make the break in the DNA. But in and of itself, it doesn't know where to make the break. The only way it knows where to make the break is it complexes with what we call this guide RNA. And the guide RNA has a sequence in its end that says, this is where you should make the break. So it tells the Cas9 where to cut. And the power of this system is that it's relatively simple. It's not quite do-it-yourself in the garage, despite the advertisements, but essentially any lab with reasonable molecular biology experience can do this. It's very active in multiple different settings, and it's highly specific when it's carefully used. So in the next few slides, I want to show how we're applying that to cure a disease called sickle cell disease. So sickle cell disease is caused by a single change in the nucleotide sequence of a single gene, the letter A converted to the letter T in a gene called the beta globin gene. That means that the cell, instead of making a glutamic acid in the protein, puts in a valine, which causes the protein in the red blood cell to, instead of stay as a what we call a tetramer, now polymerizes. And those long polymers convert that round red blood cell into a stiff sickle-shaped red blood cell, hence giving the disease its name. And the consequence, so what you can think about sickle cell disease is a beautiful human body with two broken headlights, so it really can't function very well in certain circumstances. And the manifestations of this disease are that when we get those stiff sickle red blood cells, it blocks the ability of red blood cells to get to the distal tissues. The red blood cells carry oxygen, and when you don't get oxygen to your tissues, it causes tissue damage. It can cause infarcts in the brain, it can cause problems in the kidney, but the most frequent manifestation is actually the inability of getting oxygen to the bones, causing deep, bony, painful crises. And here's a picture drawn by a teenage girl with sickle cell disease trying to convey the pain that she goes through when she has one of these crises. It also results in early mortality. So even in the US, where we have a sophisticated medical system, the median life expectancy for people with sickle cell disease is in the late 30s to mid 40s. But in Africa, where there's many more people with this disease, the average lifespan is actually only 5 to 8 years. The disease affects red blood cells, which is boxed here. Red blood cells only have a transient lifetime, so we can't try to fix red blood cells. Instead, we have to fix the somatic stem cell, which would give rise to red blood cells for the lifetime of the patient. And so ultimately then, we need to do that typographical error correction in the hematopoietic somatic stem cell. And so we'd like to convert then our car from having two broken headlights into a car that has one broken headlight. And when we use our genome editing system in stem cells from sickle cell disease patients, what we find is we get all three outcomes that I described to you earlier. About 10% of the time, we see no change in the sentence that causes sickle cell disease. About 40% of the time, we actually make the sentence even more garbled than it is. But about 50% of the time, we actually are able to correct that sentence. And so what we're doing now is taking this laboratory process and working with the FDA to develop a clinical protocol where we can begin treating patients with sickle cell disease with this Cas9 genome editing tool, hopefully to begin in 2019. Now, I work at a beautiful hospital at Stanford, Lucile Packard Children's Hospital, and Packard and Hewlett, obviously we're partners, and we're very excited to begin treating tens of patients in the US. But as I've mentioned, this disease is something that affects people around the world. And I think one of the really important ethical questions around genome editing isn't just about what should we be doing, whether somatic cells or germline, but how do we get sophisticated technologies to parts of the world that don't have the same GDPs as us. The next three slides, I just want to highlight some of the things that we should be careful that we don't conflate with each other. So the first thing is that genome editing is not CRISPR-Cas9. Genome editing is a process of changing the DNA that can be done multiple different ways. CRISPR-Cas9 is one tool that makes it easier, but it is not genome editing itself. Conversely, there are many other things you can do with the CRISPR-Cas9 complex other than doing genome editing, including what we call epigenome editing, labeling, RNA editing, and so on and so forth. And I think sometimes when I read is that people they're making these two equivalent, and they're really not equivalent. The other thing that I think we need to make sure that we don't conflate with each other is the idea of using genome editing to cure or treat a serious disease versus genome editing for enhancement, creating dystopian soldiers, or what I think is even more pernicious is the idea that you see on covers of magazines like The Economist, where you show this blonde-haired, blue-eyed baby boy, Caucasian boy, with the implication that that is somehow a perfect baby. And I think these are very, as I said, pernicious implications about the uses of genome editing. The final thing I want to make sure that we understand is that somatic cell editing to treat disease, so treating a somatic stem cell that lives in the patient, but once the patient dies, the somatic cell the editing ends with the patient, versus germline or heritable editing, where you might be modifying either the progenitor, the sperm or egg that go into making a person, or directly in the zygote itself, which again would be passed along to future generations. So being careful to distinguish between somatic cell editing and heritable or germline editing. They're not the same thing in my mind. And then finally, I just want to end with saying that I believe there's some perverse incentives on how we communicate science. And there's really well, I won't say no. At many different levels, there is an incentive to dramatize or sensationalize the discovery. As a scientist and researcher, I'm more likely to get promoted and funded if my work shows up in the New York Times. The journals are more likely to publish my work if it's sensationalized, because they're more likely to increase their revenue or status. Companies who market this are more likely to raise funds or have their stock price increase by sensationalizing or implying their work is greater than it is. And finally, journalists are more likely to get clicks and eyeballs or promoted when they sensationalize or dramatize the discoveries. So where is the check in this process? And I don't have an answer. I want to end by just thanking the number of people in my lab and I work with. Without going through all the names and all the people, I think what the major point of this slide is to point out that science is now a team activity. And while I get to stand up here and talk to you about what we're doing in the lab, this is backed up by a large group of investigators and scientists working hard to develop new transformative therapies for genetic diseases like sickle cell disease. Thank you, and I look forward to your questions.